Tuesday.
That module 2 is a lot about the lab. Are we
Yeah.
going to have a lot of lab questions on this exam? Because I watched all your mini lectures and about when is that going to be all on the exam?
A lot of the terms are, you need to know what facultative anaerobic is and stuff like that. Yes.
Okay.
Yeah.
Very good. I didn't know if you had to like know what like the chocolate plates.
Yeah. So I do have, there are going to be some questions on there, for instance, and that's why I was asking about this one media, because I wrote a note about it. So I have a note about the augers, because I did talk about those in one of the video, but one for sure is the McConkey auger is for gram negative, and McConkey, so that grows E. coli, and McConkey auger is both differential and selective. So it only allows gram negative to grow, but also gram negative can grow, and they can be pink colonies if they're a lactose fermenter, but they can be clear colonies if they're a non-lactose fermenter. So say I have a urine and I plate a urine and I plate it on a blood auger and a McConkey. The doctor calls the next day, we haven't done anything. He's like, what do you got on that urine? Well, if I see it's growing on the blood and the McConkey, I can say we've got a gram negative rod. And some doctors will even say, is it a lactose fermenter or a non-lactose fermenter? Because if it's a lactose fermenter, he's going to think E. coli. If it's a non-lactose fermenter, he might think Pseudomonas. So those augers that are differential are very, very helpful. The other auger that is selective but not differential is PEA. And PEA is selective for gram-positive organisms. So gram-negative don't grow on that.
What did you say non-lactose was?
Clear.
What? No, what is it?
Oh, Pseudomonas. Pseudomonas, yeah. That's a common UTI in hospitalized patients. And then the other one that I wanted to mention that's in your notes, but it's not going to be in your textbook, but you can just Google it, but it's Martin-Lewis auger. That's going to be in your notes, but I couldn't find it in your textbook. But in my previous notes, I had the students refer to their atlas, and see, I forgot you guys don't have an atlas. So the Martin-Lewis auger is selective for Neisseria gonorrhea. Martin-Lewis auger is selective for Neisseria gonorrhea. So that, because think about a vaginal swab, it's going to have possible yeast, it's going to have bacteria, it could have possible contamination from stool. And so the Martin-Lewis auger inhibits yeast and a whole bunch of different things. It has a lot of inhibitory agents in that auger and only allows the Neisseria gonorrhea to grow. So those are some of the important augers that might be in on the exam. Okay, questions?
Okay, so my cultures grew, but they're not, I don't know if you can see that, but I feel like I remember them being like more in here.
Do you have sediment?
Cloudy.
Do you have sediment on the bottom?
A little bit, but not a ton.
Okay, is that the Saccharomyces?
No.
The Saccharomyces, it took 3 days for mine to grow. The Saccharomyces likes it warm.
Yeast grew.
What's that? Which
yeast.
one is? That's the yeast?
Yeah.
Wow, that grew a lot better than mine. Well,
this is the staph. Staph is clear. And then the E. coli is like,
yeah, that's growing.
Cloudy.
Yeah, that'll be fine. How many hours incubation is that?
That's 3.
3 days?
The 24 hours.
So it's only been 24 hours.
24, yeah. Yeah,
oh,
24, yeah.
Oh, yeah, you got to let those go. Let them go 48.
Okay.
Yeah. Does the staph have any turbidity at all? Just give it a little shake.
A little bit.
Yeah, let that go another day. And your temperature's okay? You're at least 24 degrees? in there? Okay. Okay, let that go a full 48 hours. I mean, you can go ahead and do your streak plates of the other two. They're plenty growing. If you want to streak out your E. coli and your Saccharomyces, the yeast, you can do that. But let that staff grow.
Got it. And then we're supposed to, for the blue things, we're just dipping them in alcohol?
Yes, let them sit in the alcohol. It doesn't tell you how long, but I let mine stay in there like 30 seconds. What were you saying, Kathy?
I'm saying that it's been cold here. You're used to warm, but it's been cold here. So room temperature has not really been, at least I don't know about anybody else, but we're not really heating.
Yeah. I know. I'm in Massachusetts now, and I am freezing. So anyway, I know they do have to be warm. So you're going to have to, and the cooler it is in your house, the longer it's going to take for them to grow. So try to keep them warm. All right. Other questions?
So when we go to pour them in the dishes, are we doing that tomorrow with Kathy? Or should we already do it by ourselves?
No, you can do them with me tomorrow.
Okay. Because I was kind of confused on like how much, like should we dump the whole thing in? Or
No. Sam, it's 2 plates per 1 broth. So only pour half of it.
Okay.
2 plates per 1 broth.
Okay, I'm confused. Sam, are you talking about streaking the plates or pouring the plates?
No, when the E. coli after it's all done culturing.
Yep. And last week we made plates, right? We poured the plates. So if you were with me and we poured the plates, now we're talking about streaking them, and we'll do that tomorrow. And you're gonna take just a little bit of your sample.
You just dip in with your loop. That's it. Don't pour anything.
Okay.
Yeah, you dip with your loop.
Okay.
Any other questions?
Are we getting on tomorrow at 9:30?
Yes, 9:30 tomorrow. I experimented too with the little swivel patty and trying to use the swivel, but I can't do it on my phone because I don't have enough. So anyway, I think I got it figured out. So hopefully we'll have a little better video anyway tomorrow so you can see a little better, but keep your fingers crossed.
Yes, and I just wanted everyone to make sure they understand that when Kathy does that, that's not just open to Kathy's lab, that's open to anybody's lab. And anybody, can just move at their own pace. If you're comfortable with just looking at my videos and moving forward, that's fine too. So my lab can watch Kathy. We're all just one big group here. So just do whatever you need to do to be able to get through this. I see that most of my students, I only have just a couple that haven't finished their prereqs. So hopefully, they're due, Wednesday this week. All those prereqs, all 4 of them need to be in by Wednesday. And you should be starting that lesson 1, which, what was I going to say? Kathy, you have 1 student. Do you know if she's gotten her kit yet? Christy Hamblin?
I don't know. They were supposed to be sent out yesterday.
Oh, good.
So I did tell her that I'll give her a little extra time with the first lunch, but she's all set to go as soon as she gets it. She's done her prereqs.
Okay. So like I said last week, the first couple labs are still a little bit challenging. Once you get to carbohydrate fermentation, motility, those are going to be a breeze for you. Well, maybe not a breeze, but they're going to be easier. The other thing is that when you get to the morphology and the staining, no, you're not going to get to look at your own slides because we don't have scopes. But I want you to go through the process of making those smears and doing a gram stain. And you'll see in the procedure, it has you make your smear, sit around and wait for it to air dry, then heat fix it. I made all my smears, sat around and watched the 3 of them air dry, and heat fixed all 3 of them at the same time. But then your staining tray, at least mine, was only big enough to do 1 at a time for the gram stain. So, which is fine, because then you can just go through the process 3 times, which I really like, because you might have some questions on the exam that are applied questions. So like the gram stain, you put your crystal violet on. The gram stain is a differential stain. It differentiates between gram positive and gram negative. So the crystal violet goes on and stains all the cells. If you stop the procedure then, everything would be purple. Then you put the mordant on. The mordant is like a seal. It's the iodine. Iodine seals the crystal violet in the peptidoglycan, in the cell wall. And then you rinse after every step, rinse with your water, just tap water. Then the alcohol, you just gently tilt your slide and let it drip down the slide. Then rinse it real quick with water. If we looked at the slide right then, because I had a student do this one year with their unknown, they totally forgot the safranin, which is a counter stain. They totally forgot their safranin, the counter stain. And they couldn't see anything on their slide at all. That was because they had a gram negative organism that wasn't colored at all because they forgot the counter stain. So they stained them with with the crystal violet, but then it was decolorized, and because they didn't counter stain, the cells were clear because they were gram negative. Another thing that a student did with their unknown was, and these are all things that can happen with a gram stain, they forgot the mordant. They forgot the iodine step. And so without the iodine, they were having gram negative cocci because they didn't use the mordant. It didn't seal the crystal violet in the cell. And so their gram positive cells were appearing gram negative because they forgot the iodine. So they decolorized and then they restained with the pink. So you need to know that saffron can stain any cell that's unstained. It doesn't just stain gram negative. It's just that it doesn't stain the gram positive because they're already stained. It won't stain over that. But if you forget to add the mordant, then they're going to be pink. And so we've seen that happen twice with student unknowns where they've messed up because it's a very specific step-by-step process. So I'm glad you're actually doing it 3 times. At least then you can troubleshoot questions and stuff like that if you see those. But like I said, you're going to go through the procedures, but you're not going to actually look at them under the scope. You're going to go up to the V-scope library and get your gram stains from there. Okay. Questions?
So on the schedule, there's beginning data research for unknown. What is that? I couldn't find it anywhere.
Yeah, I haven't posted it yet because I didn't want to freak you out, anybody. So.
Oh yeah, it says it starts today, so I wasn't sure.
Okay.
Get it going.
So I'll put it up, but I don't want you to I have the due date in 2 weeks, and I know this beginning's a little bit overwhelming. But if you're just sitting there watching TV and you're not studying micro, it could be something you could work on. So I will post that today. And what it is, it's going to be a sheet of all the bacteria. I think we're giving you 20 instead of 50. The regulars in the past, we give 50. And I just got to go get my plug real quick because my battery's dying. I'll be right back. Kathy can answer some other questions. I'll come back to that.
Is it going to be roughly about the same as what it would be in class as far as the unknown? We have to have our little book, or is it all going to be pretty much an HOL?
It's not going to be an HOL. You're going to do research based on the table that Patty's going to post. So she's going to post that table and you're going to fill it out. You're going to look for all the results for those 20 organisms and you'll put it in the table. Then you're going to be doing an identification based on the all that information that you've looked up. Okay, I think we're going to have you, well, good, Patty's back because she can answer whether, I think we were going to have you make a dichotomous key or a flow
Yes.
chart.
Yep.
And you'll use that flow chart. You'll use the all the tests you look up on the sheet she's giving you to make your flow chart, and then we'll give you organisms virtually through a PowerPoint, and it will show you results. It'll give you a case study, and you'll use those results to go through your dichotomous key and identify the organism that we've given you.
Y'all get different organisms.
So
There'll be
our
20 different organisms, 20 different options. that
yeah,
you
and
will
I
get.
think you have 12 tests to look up. So we're having you do all the tests you're doing, you know, like motility, DP, and stuff like that. So they're going to be tests you're doing, but there's a couple tests that I need in the data that you're not doing, but I've added them to the wording in the exploration of the lab. So something like citrate. You're not doing a citrate, but I want that citrate on the chart. So I put the wording in the exploration part. So today I'll post a chart. It's going to have the 20 organisms, the 12 tests to look up. You can use your textbook, but also, and we can take the time right now and I can show you how to use those books that are in the library. We can do that. I'll share my screen, or Kathy can, because we can show you how to look stuff up, because I did the chart. I was in Florida when I looked up all the data, so I had nothing. I didn't have any books from the college. So I just did all of mine from the books that are in the library resource in Blackboard. And I found that I downloaded the whole book. I have the whole book on my desktop. I just found that was easier. I think I have 3 books on my desktop that I downloaded, and I found all the results there. The ones when you complete it, there were a couple out of the whole data table that I couldn't find. And just put, if you can't find it at all, just put NA, not applicable, that you couldn't find it, because I did have a couple of those. But I look, so you're going to look up all that data and try to have it done in 2 weeks time for that. And then I'll give you a little extra time then beyond that to do the keys, the dichotomous keys. Now, what I ended up doing, I had a key that started, I initially started with a key that was the gram stain, and then I had gram-positive cocci, gram-negative cocci, gram-positive rods, gram-negative rods. And that is a good way to do it. It just ended up being a big thing. You need like a poster board to do it that way, but it is very clear. But you'll have to take a picture of it and send it to me, because you do have to, and to Kathy, you do have to submit the data table and the dichotomous keys. But I ended up doing one for gram-negative rods, but I think I divide mine in gram-negative and gram-positive. There's no right answer. There's lots of different keys. I developed them different ways. So there's no one answer for those keys, the dichotomous keys. We just want you to see, okay, if I have a gram-positive cocci, what's my next best test that I should do to try to distinguish these 4 gram-positive cocci that are on my chart? Okay, so once you have the data done and the dichotomous keys, then what happens is, at this point, we plan to give each of our students a gram-positive unknown and a gram-negative unknown. And it is just a PowerPoint presentation that has 10 slides. The first slide just gives you an opening. It's a case study. And then we just show you pictures of the results. You have to interpret the results correctly, and then figure out from your dichotomous key. So most of the work is looking, and it's true on campus. If you were doing the unknown on campus, most of your time is spent researching. It's very little time setting the stuff up and reading it. So most of it is doing the research and trying to figure out what's the next best test. In your case, I'm giving you the test and the organisms, you make the key, and the actual identification of the organism when you get your PowerPoint that we'll send you, that's going to be easy. And it's going to be worth less points than the data table and the keys, the dichotomous keys. So, because that's where the work is for that. So I will post that today. And does anybody have any questions before we might share the screen to show you how to look that stuff up?
Oh, my only question was going to be like, could we see an example or something of that? Because my brain is just lost from all that information.
I'll share. Let me just share real. Oh, I can't share. Kathy, can you share this? Unmute your mic there just for a sec, Kath.
You know, I can share mine. I don't know why you can't share it this time, why I can't allow you to, because I did.
Okay, I'll try. I'll show you what I'm going to post today and what you need to do.
But yeah, I'm not, it doesn't look like I'm able to though.
So you said we have to figure out what the next test we would run.
No. Unfortunately, you will not have that experience online.
I didn't know if we had to figure it out and then you would give us the next test, even if it was wrong, so that we can try to like
Figure it out.
Yeah.
No, but what you can do, if you can't get an answer from the slides that you gave us, or you're not sure, you think it's this, but you want another test, in that case, you can email us and say, you know, I think it's this, but I'm not 100% sure that it's not this. Could I see what the result would be of this particular test? Yes, you can do that. We don't have any problem with that.
I think you can share now, Patty. Can you share? I just made you a co-host.
Okay,
I figured it out.
I'm just trying to find my data sheet that I'm going to post here real quick. What did I title it? Hold on just one second. You guys can ask questions to Kathy while I'm trying to find, oh, here it is.
So we're streaking plates tomorrow. Are we going to start lab 2 then tomorrow as well?
Yeah, I will show you how to make the slides and stain them tomorrow too. So for those of you who want to stay a little bit ahead.
Okay, so the whole doesn't unlock the next one unless we submitted the one before. So is there any way you can help us see the instructions to unlock the next lesson on the whole so we can see the instructions for the next lab?
Oh my gosh, I can't believe that.
We can, I think we can unlock it though, Patty, can't we? I think.
Yeah, but I don't know if they can go on without, I think, well, there's a couple adjustments, Kathy. We can unlock all the labs and we can unclick that they have to do them sequentially. And I think that should allow them to see them as long as they don't have to do them sequentially.
The only thing I'm worried about with that, Patty, is we still have a couple that are not done the prereqs. Is that going to influence whether
No,
they?
that should,
Okay, so,
no, they can't move forward at all without those.
all right.
And is my screen being shared?
Yep.
Okay, so these are your organisms. So there are, actually there's not quite 20. So these are the list of your organisms. Then what you're gonna do in the next couple weeks, morph and gram means. So let's see if you know some of these. Like all your staphylococcuses, or let's do E. coli, because I've already said that today. What is E. coli as far as morphology and gram? Anybody? What's the morphology of E. coli? So this is a rod, and it is gram negative. So you're putting the morphology, is it a rod or a cocci? I will tell you right now, none of these are spirillums. Those are the 3 morphologies, rod, cocci, or spirillum. I did not put any spirillums on the list. So the morph is if it's a rod or cocci. Some of them are going to say they're Cocobacillus, so you can just put CB, but a Cocobacillus is a rod. Then the gram is either pos or neg. So let's go down here. Staph is cocci, and it is gram what? Positive. Those are gram positive cocci. Aerotolerance, you're learning in lab. Well, no, I did put some stuff in lab that I added, but aerotolerance is it facultative anaerobic? Like E. coli is facultative anaerobic. That means it can grow in the presence or the absence of oxygen. So really the only options you're going to have in this slot, I didn't give you any anaerobic organisms. So the only thing you'll have in this slot is either facultative anaerobic, FA, or A for a strict aerobe. You shouldn't have any that are anaerobes. They're either going to be a strict aerobe or a facultative anaerobic. Catalase is pos or neg. You're going to do a catalase in your lab. Oxidase is positive or negative. You're not going to do that in the lab, but I put the procedure in your lab so that you can interpret the slide pictures that we give you. Motility you do in the lab. My motility did not work that great. Motility means you're going to straighten a paper clip, or hopefully somebody's figured out something to have something straighter that you can sterilize, or not sterilize, but disinfect. My motility didn't work that great. I don't know if I moved the paper clip or because it was bent, but when you set up a motility, you go straight down your tube and you come back out the same path. And the thing is, if it only grows in that pathway, it's not motile. If you can see your streak line, it's not motile. If you have growth going all the way out to the sides or even out half your tube, it's motile. It's growing past where you inoculated it. So motility will be positive or negative. Then glucose utilization. Is glucose fermentatively used, or is it only oxidatively used, or if it's just totally doesn't use it fermentatively or oxidatively? We normally just teach the students fermentation, but there's an organism on here that's negative for glucose fermentation, but it is positive for glucose oxidatively because it's a strict aerobe. Mannitol and xylose are sugars. You're just going to put positive or negative. Those are both sugars. Indol is positive or neg. VP and citrate are positive or negative. You're going to be doing, I can't remember, do they do indol, Kathy, in lab? I don't think so.
I don't think they do.
I added it though. And then you're going to do VP in lab. So those are the tests. So it's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 tests. I had a column. I filled my column out. I put the book and the page number I was finding the information on under reference, so that if I quickly wanted to go double check, say, Proteus vulgaris, I would go, okay, Proteus vulgaris, I found those results in this book on this page. It just, if you go back, it makes it easier that you referenced it there. So you're going to fill this out, and I would like you to put your page number in book. And then once this is completed, you will submit this. And I can't remember, but it's somewhere around 30 points of the 60, because this is the bulk of the work here. So I'm going to say it's due in 2 weeks, but we might have to extend it 3 depending on how everything's going. But we only have, 6 weeks to do this, so I don't want to extend it too far. But I would say try to get it done in 2 weeks, but most likely we may have to give you 3. We'll just see how it goes.
And we can look all this stuff up in the books, correct?
It's
So is
all
that-
on, yep, so it's all, Kathy, do you want, oh, I can share now. Hold on. I will show you right now. Let me go into Blackboard. Okay, so now, can you see my screen? I'm going to go into library references. And then here is the Burgess Manual Systematic Part 2. So if you go down, these are going to be like your E. coli. But if you go down, I downloaded, I believe, I didn't download this one or this one, but I did download this Manual of Clinical Microbiology. and then I downloaded this one, and this one. So I think I downloaded 4 of them, but mine are downloaded so I can show you already what it's going to look like. But this is where you find all your reference books. This is a good one too, this Manual of Clinical Microbiology. The other thing is, oh, Kathy, did I put that in here yet? If you click on laboratory, oh, Kathy, this online Bergey's help, I think I have the updated one. Oh no, you were going to update it for some of our new organisms.
I haven't put the new organisms on it.
Okay, well,
So-
I'll make this available, guys.
Okay.
I'll
I'll
make this-
update it when I get the new organisms on it, but
Okay.
they can use it for now.
For now, you can use that. It tells you which book, which organism is in. So I'll make that available for you right now. And then once we get the other organisms on it, once we get the other organisms on it, we'll give you that. Let me just, and then I'll just go very quickly to the book, to one of the books, to show you how to use them. I don't even remember where I put it. I think I put it in MicroLab. Right. So here's one of my books. Here's another book. These are all my books I've downloaded. Okay, so this is Bergey's. This is one of them that is, so basically what I do is you can come down, you can do one of 2 things here. In Kathy's book, you see where my cursor is up here by the page number? You can actually put the page number that Kathy gave you, and it'll get you close to it in there. You type the page number there that she gave you, and it'll bring you close. The other thing you can do is go to the back, and I gotta increase my magnification here because I can't see it. So let's take Escherichia coli. So this is the index, and I'm going to show you very quickly in the index. If I go to Escherichia coli, well, here, see this Enterobacteriaceae? That's the whole family of a bunch of, quite a few of them that are on your list. That's a family. E. coli is in that family. Proteus vulgaris is in that family. Shigella is in that family. And do you see there's one page number that's in bold? 587 is in bold. So now what I'm going to do is I come up here, and if I type 587, it's going to bring me close to where I should be, because sometimes what I put in here, if I find, where's my page? So I'm only on 569. I don't know why it didn't take me. It's not taking me, but at least it's bringing me to about the right place. So 587, 579, these online books are kind of a pain. So here's my family, Enterobacteriaceae. This starts with the family. They're all gram-negative rods. Where you have to be careful about, motile, see where it says motile by peritrichus. Peritrichus, you learned in chapter 3, means flagella all over the surface. Motile by peritrichus flagella, except for tautomella or non-motile. So make sure you get to the end of the sentence. So it is motile or non-motile. So there's going to be a difference in that. grow in the presence and absence of oxygen. So what are they? What's their aerotolerance? If they can grow in the presence and absence of oxygen, they are facultative anaerobic. So they're all facultative anaerobic. So what it does is it'll go through and describe the whole family. Then if you come down further, and I'm going to get you, if we get to here, this is the chart you're going to be dealing with, okay? Indole, methyl red, Voges, Proscar, and citrate. I talk a lot about that in the notes and the lab. That's called the IMVIC. IMVIC. Indole, methyl red, Voges, Proscar, and citrate are the 4 primary tests used to identify Enterobacteriaceae. So let's say we're looking at Citrobacter frundii. Citrobacter frundii. Well, let's look at Citrobacter almoninicus first. Its IMVIC would be POS, POS, neg, POS, which means it's indole positive, 100% are indole POS, 100% are methyl red POS, 0% are VP POS, and 95% are citrate POS. So on your chart, if you have Citrobacter, which you don't, you would put POS for indole, POS for methyl red, negative for VP, and positive for citrate. The problem is if you have a 33, what you would do is put a V for variable, and then put parentheses 33, meaning it's too iffy. Only 33% are positive. The question comes into, well, what about like 87, 60, stuff like that? My personal cutoff is 85. So if it's 85 or above, I tend to say it's positive. If you're more comfortable with 90% and above, so 85 would be variable, but 95 would be positive, you can use your own breakpoints. And then on the other end, if it's 10 or lower, I call it negative. But anything, so basically 10 to 90 is what I call variable. 10 to 90 is variable. Anything that's 93 or whatever I would say is positive. Anything that's say 5 I would say is negative. That means only 5% are positive. So that's how I do it. So there's charts here. So you look stuff up on the charts. But then the other thing that you can do is it gives you all these charts, which you're going to find, I think, at least 4 of them on those charts. But the other thing you can do then is you come down and then you start at the genus level. So I think the first genus that they talk about is E. coli. And so right here is the beginning of genus. So we went from family to genus. So now this discussion, because you want to keep your eye on this stuff, you're moving from family to genus now. And so now you're getting information. Oxidase negative, modal by peritrichus. These are all these are all your Escherichia genuses. Okay? And then obviously if you move down further, then you can see the Escherichia coli. They'll specifically talk about the species coli. So you just have to keep going deeper and deeper into the text on that, for that.
Are we allowed to print this table?
I don't know if it try it. I wouldn't print the whole book, that's for sure.
No, just the table for the different
you can try it.
organisms you want.
I think that would be really handy. I had a hard time using the computer because it goes to the next page but doesn't take the test with it. So if you can print it, it would be awesome. Try it. I don't know if it will or not. I'm assuming it would, but I don't know if there's some control on it. Okay, so that's just what the Burgess looks like. But again, the page number you can type here, go to the index to try to find your organism, and go to the bolded one first. The bolded page number. will take you to where most of the information is. Okay, questions. We're down to only 10 students here, but questions, anybody? Okay, so I guess I have another question. Should I schedule something for Monday just for last minute questions about the exam? Or are we good to go? Because I won't see you now till the exam on Tuesday, because Kathy will host tomorrow.
But so am I getting on with you in the evening time to show that I am who I am taking this exam?
Yep, because I have another student that has to take it at 6 o'clock. We're planning on 6. 6 o'clock on Tuesday.
Collaborate or Zoom? Zoom time's out.
I think we can do it on Collaborate because there's only 2 of you.
Yeah, you'll want to make sure
Let's just do it on Collaborate because there's only 2 of you. The
right.
problem is on Collaborate, I only see like 3 of you at once. So.
And you're gonna sit on while we take the whole exam then?
Yeah, I just have to make sure it's you.
Okay.
Taking it. And that's why I had you guys upload your picture ID so that I could see. That's the hard thing when we never actually see you, that I know that I'm dealing with who I'm dealing with. So that's why I had you give me your picture ID. But yeah, so I'm just going to be in and out, but it's mainly just to identify that it's you.
Patty, I had several students who didn't get that message before they started. Do you want me to specifically get back to those students? I didn't give them a point for that requirement, but I haven't gone back and asked them to resubmit it.
What are you talking about? I'm not certain.
Remember, the requirement on the prereq was that picture ID. And I
Yes,
have several people.
All my students that didn't do that, I emailed them and said they have to do it.
All right, I'll do that then.
Yep. Okay, I guess for now, unless I have requests, because the thing is the next module stuff isn't as technical as that. I was worried about chapter 3, you know, with the cells and stuff, and there was quite a bit about the scientists and stuff, but the other stuff is a lot of the lab stuff for that. So I think we'll be okay with that. If I got a lot of requests that they want to just meet on Monday, I'll do that. Otherwise, I will see everybody on Tuesday for the exam. And that I'm going to have us get in there at like 9:15 or 15 minutes before. Christine, yours would be 5:45, just so we can get everybody set up and ready to take that. Okay. So remember, that's also, that module includes chapter 19, the gram-positive module that opened today. And again, I know some of you have asked, again, I don't open the mastering until we're 2 weeks out, because I don't like, I don't agree with students trying to get all their assignments done on the free period. So the free period end, I think, after 2 weeks, and then after 2 weeks, I'll open up the rest of the assignments for students to do in mastering for that. So they will get opened up, but they're all opened up now for this module.
So we're looking at chapter 19 too for this first test?
That's the gram-positive, right?
I believe so.
Yeah.
Okay.
Yeah. So that's on that module overview.
Yeah, okay.
Yeah. Okay, and like I said, email Kathy or I with questions. And I mean, I think everybody, considering that we're stumbling with technology, everyone's doing really well. So I'm very pleased so far. And I was so happy, out of 40 students, 38 had everything done for module 1, including the quiz. So I was like, I got up this morning. I'm like, woo, awesome.
Good start.
It is a good start. So I was very happy. I mean, I'm stressed because this technology is just driving me nuts. But it's just getting, I don't know what happened when I shared my screen in Collaborate that I lost my sound.
You know, that happened to me once, and I can't remember why.
Because I'll ask Dean about that, why that happened. But
If I
anyway.
remember, I'll let you know. I did something specific, and I was still able to share my screen after that. There was something about it that I had done.
Okay.
And I can't remember what it was, but
Okay.
it happened once. And I learned my lesson.
And also, all of you, I mean, I'll obviously send a message, but at some point I might be unavailable for a little bit because I'm on call right now. That's why I'm in Massachusetts. My daughter is 38 weeks pregnant, and she has a 2-year-old. So that's why Grandma is here.
We have guided notes for chapter 19.
Yep.
Okay.
Yep. They're all in the packet in the back of that.
I have to print those ones off. I thought that this big old packet was what we had for the exam, so it's 37 pages long.
For module 1 and 2.
No, just module 1. Because I went above.
Oh, because you typed it.
I did.
Oh, that makes it so much longer.
It does, but my hand hurt after like 3 pages. So I was like, this isn't happening anymore. I'm not doing this.
Yeah, because all the notes I try to keep about 18 pages, which is about the amount of notes they would take in class in a given, week. So, okay, guys, good luck. Make sure you email us if you have any questions or any problems at all. You're doing great. You're really doing well.
I'll see whoever wants to tomorrow morning for lab at 9:30. And I will send out, I haven't decided because I recorded this. I've got to figure out how long it takes me to download it
Right.
to YouTube. And I have a feeling if it takes too long, I'd be better off in Blackboard tomorrow.
I know.
So I've got to work out this technology too. So I'll let you know either tonight or tomorrow morning where we'll meet.
Yeah,
Can we use those little clear cups to do our whole gram stain thing, or are those meant for something else?
they are meant for something later in the lab. Yeah.
But
All of those, the big package of cups?
Yeah, but you should have got a little
you got a little, it looks like a plant.
bottom stand.
Yeah.
Oh,
That's your staining
A
tray.
water collector underneath you.
is it like a plastic little
Yeah.
lid looking thing?
Yeah.
Okay.
Yeah. Looks like a coffee lid.
Okay,
Yeah, that's your staining rack.
got it.
The other thing is too, is that, what was I going to say? Oh, so I don't even know. I don't think we got that review session recorded then, but
No, sorry.
sorry. It's just, we can't, the thing is that with so many people using YouTube and stuff now, like those videos I did of myself, the longest one is 15 minutes, but even like the 10-minute one, it was taking me 4 hours to upload those to YouTube.
Yeah.
Everything's just so clogged up, So, I mean, an hour and a half review session, I don't know, that would have taken me 2 days to upload to YouTube.
The last time I had some long ones, it also booted them. It'd say you've timed out.
Yeah.
So I'm
No.
concerned about just this one as well to see how long I can, I don't know. So we'll just have to see.
Okay.
But that made me think of something which now has gone out of my mind. So never mind.
All right.
It's tough to get old, you guys. You're all so young. Just wait.
All right, you guys, good luck. Make sure you email us if you have any issues.
All
Thank
right,
you.
and I'll see you guys tomorrow. Have a good day.
You too.
Thank you. Have a good day.
Bye, guys.
Thank you.